really familiar term. What are they? Yeah, they carry meiosis. They have all of the same genes, but potentially different versions of them, right? Which is what this next kind of part of the unit is going to be all about. Because we're getting into our genetics unit, and so hopefully very familiar territory in notes today and tomorrow. And your homologous chromosomes, yeah, half of them come from your mom, right? One half of the pair comes from mom, the other half of the pair comes from dad. So we call those different versions of genes alleles. So we've already used some of the terminology that we're going to go over today. And again, just kind of to recap, these are homologous chromosomes before replication. These are homologous chromosomes after replication. So that's when our sister chromatids show up, right? After replication happens. We have those identical copies that are attached to each other. They cross over during meiosis, right, et cetera, all that good stuff we talked about last week. So today we're getting into Mendel. We're going to talk a little bit about his more simpler experiments today with mono hybrid crosses. We got a lot of vocab, so all these words, are they kind of bringing up some memories of past biology experiences?
Oh my gosh, I did something with my brother on this yesterday.
Did you?
It was like the hexagon puzzle or something.
Well, I haven't seen that one, but
Maybe not a hexagon.
what kind of vocab thing?
It's like triangles you have to match up together. And they're like into question.
Oh, I remember.
Yeah.
I don't remember what it was, but I remember that.
It took me like 45 minutes.
Sounds like some kind of vocab exercise. So lots of vocab that we're going to go over today. Genotype, phenotype, homozygous, heterozygous, recessive, dominant, etc. So I do have a tendency to move kind of quickly through this stuff because so many people have seen it. It's like the third time you've seen some of this information. But if you need me to slow down at any point, holler. So this gentleman here, Gregor Mendel, father of genetics. You will know him as the grower of many, many types of pea plants. So we'll talk about Mendel and peas today.
Yeah.
Pea plant grower.
You want me to read it?
This is just a quick kind of history lesson. I'm not going to expose you over this, but he was a monk in Austria. Born to a pretty poor family, so they grew a lot of their own food, and because of that, he kind of had a basic understanding of agriculture and plant breeding, etc. He ended up studying math and science at the University of Vienna. Vienna, Austria.
Yeah, Indiana.
Like, dude, that's cool.
And so I think partially because of his background and that he knew how to grow plants, he got put in charge of the garden. And as he was growing these plants, he kept really good records and did some various experiments, specifically looking at various different crossings of traits. So we're gonna look at some of his data today. So basically, figured out the basic tenets of genetics without even knowing about DNA. So just kind of keep that in mind. No idea what DNA is. No idea what chromosomes are. Just based on really careful research, was able to figure out some basic rules of genetics. So kind of impressive. Just a little impressive. However, sadly, that work got ignored for a really long time. But today we definitely appreciate his genius. Like I said, pretty impressive. So some background on pea plants, if you didn't know, this is a pea flower. Most flowers have both a female part and the male part. So the pistil is the female part, and the stamen with the pollen on it is the male part. And so the way that he would control these pollinations is that he would cut off the pollen so that it wouldn't attach to the pistil, the female part. And then he you could cross-pollinate plants. So you could take pollen from one plant and paint it on the pistil of the other of the flower. So pea plants are one of these plants that will self-pollinate. You know what that means? So if he doesn't cut off the pollen, the pollen portions of this, they're going to fertilize the flower themselves. Okay, so you have the option to either cross-pollinate, to take pollen from another plant, or you can self-pollinate. That is actually kind of an important part of his questions on that? So Mendel followed a very basic process for all of his experiments, all his genetic experiments. So he was breeding these pea plants, and the first step to all of these experiments was to cross-pollinate through breeding parents. So let's just make sure we know what he was doing here. What did he think cross-pollinate means? Yeah, so you take the pollen from one flower and pollinating another flower. Through breeding parents, what do you think that means? You have a true breeding white flower, what do you think that would generally mean? Well, if it's self-pollinated, all of its offspring would be white. And if this purple flower self-pollinated, all of its offspring would be purple. Okay, so again, pea plants can will generally self-pollinate. So you know, if you would grow these strains so that you only got white flowers, and you grow another strain that only produces purple flowers. And then for this parent generation. Okay. So you all can see the word abbreviation P, which stands for parental. Okay, so that's how we started all of the experiments, that parental generation. Okay. Pure breeding. And see, this would take a little bit of time, right? You gotta wait a growing season for the peas to form, and then you have to plant those peas to get what we call the F1 generation. F stands for filial, which just means offspring. So this is the first generation of offspring, the F1. This example, you'll see that all of the F1 generation, all the flowers are purple. He would then let those self-pollinate to get to the F2 generation. And what do you think F2 is just the second filial generation, so the second generation. So this is the way he did it. So we started with the parents, pollinated them through the F1 generation, let those self-pollinate, and analyze the F2 population. F1 population is pretty easy to observe because they all have the same traits. I know this is super tiny, especially for those of you that are in this awkward, like, half of the classroom. But he did this not just for one trait, but for 7 pea traits. So you can see here's the parents that he crossed. So brown peas and wrinkled peas, yellow peas and green peas, brown pods, green pods, yellow pods, purple flowers, white flowers. Axial versus terminal. So axial means the flowers are coming on the side, terminal means at the end. Tall and short, tall and short. Take a look at the F1 results and the F2 results in the ratio here. What patterns do you notice in your group?
So like, I don't know, look at this, like, really funny. F1 generations, all round, all yellow. F2, mostly all round, mostly all yellow, but there's still some outliers. That's why the ratio is that.
Okay, so first of all, if you look at the F1 generation, what's always true?
They're all something.
They all show being just one trait, right? So one trait disappears completely. Kind of strange, right? When you take a look at the F2 generation, the ratios are almost always exactly what?
Ones.
Well, 3 to 1, 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 3 to 1.
Double the 60.
It's always really important to remember that in genetics we're dealing with probabilities, so there's a likelihood of percentages happening. However, everything ends up being a little bit unique. So it's not going to ever be, almost never will be exactly the expected 3 to 1 ratio, but it's going to be really close. So we're going to take a closer look at those flower color experiment. This is also on that handout I gave you, so if you want to fill in the blank squares on that as we look through this. And you can replicate this diagram with any one of those other 7 traits, right? And you see the same patterns happening. So we're starting off with the P generation. Again, P means parental. So we're taking the pollen from one true breeding flower plant, in this case purple, and crossing it with a white flower. It is notable that Mendel did both directions. So he would take the pollen from purple and fertilize white, or take the pollen from white and fertilize purple. So we did go in both directions. Talk later about why. And of course, what did he get in the F1? All purple, 100% purple. What's F stand for again? Filial offspring, the first generation of offspring. Then what did Mendel do? He let those F1 plants self-pollinate, and he ended up getting the F2. And that ratio was very close to 3 to 1. Okay, so about 75% of that F2 generation is purple. About 25% of that F2 generation is white. So 3 out of 4 purple, 1 out of 4 white. And like I said, you can substitute any trait in here. Green peas and yellow peas, all green, 3 green to 1 yellow. This repeated every single trait that he tried. So we're seeing the same ratios every time. Also, again, we see one trait getting mass and then reappearing again in that F2 generation. So here's some important ramifications of that data. Again, Mendel didn't know anything about chromosomes, but we do, so we can kind of integrate our knowledge into his work. First finding that traits come in discrete alternative versions. So individual alternative versions, and we call those individual alternative versions alleles. Right? So we talk about having the dominant allele or the recessive allele. So in most cases, These alleles are almost identical DNA sequences, so almost the same sequence of nucleotides, but with at least one difference. So you switch an A with a T, or a C with a D, or an A with a D, or a C with a C, or you delete a base, you add a base, and all those different mutations that could happen. That would make one nucleotide sequence slightly different from the other nucleotide sequence. But we still find them on the same location on homologous chromosomes. So the location, we call the term for that is the same locus. So if we were to measure out the length of this chromosome, these alleles would be at the same location on each homologous chromosome. And I actually think this is a pretty important diagram to be able to comprehend. So we're looking at two homologous chromosomes here, right? One homologous chromosome contains the allele for purple flowers. The other one contains the allele for white flowers. Questions on that? Yeah, I know this, hopefully it's pretty familiar stuff. This is just one of that diagrams. So again, the purple flower allele and the white flower allele are two different DNA variations, so slightly different sequence of DNA bases at the same spot on homologous chromosomes. Different versions of a gene, same location on homologous chromosomes. So same thing, multiple times. You probably don't very both of those yet. So, you could potentially have homologous chromosomes with both purple flower alleles, or you could have one with purple and one with white, or you could have two white alleles, right, on either chromosome. So here's a set of questions here for you really quickly. Talk about those here really quick. This is basically the do now over again, right? So we're diploid, which means that we have two sets of chromosomes. One came from mom, the other one came from dad. We call those homologous chromosomes, right? So it's kind of like having two editions of information, right? One from mom, one from dad. They're going to be similar, but they're going to have slight differences. Okay, so they're comparing them to two different encyclopedias here. Okay. Sorry, this question is so low to the board. It says, what are the advantages of being diploid? So take a couple seconds to think about that in your group. What's an advantage to having two copies?
Yeah, that's what I always say. Quality over quantities. But I did not get a response.
Okay. There's a couple good things there. First of all, genetic variation. Yes, having two copies is going to give us some differences. The other important thing I heard, if one of them's broken, you're more likely to have a copy that works, right? So basically you have a backup copy of every single gene in your body, right? So you have two copies of each gene. If one's broken, the other one might work. Both of them might be broken, but that's a lot. When we get a lot of problems is when both of them are broken. So, finding number 2. Some traits mask others. When we mix that purple and white flower, we don't get white purple, we get purple. So, that purple allele tends to mask the white allele. So even if you have both, if you have a purple and a white, you only show the purple. You don't show a combination of the two. Sometimes you do, depending on the trait. With Mendelian stuff, you don't. We call that trait that does the masking the dominant allele. The dominant phenotype. So, this might be a little bit of an extra layer that you haven't focused on before, so I think this is really important. So, if you're falling asleep on me, try to wake up. Generally speaking, this is not always true, but generally speaking, the dominant allele is going to make a functional protein. So in the case of our flower color, the purple allele codes for an enzyme that results in purple pigment. Therefore, the flower is purple. It's going to mask any allele that does not function. So this is our working copy. One copy is broken, the other copy still works. It's going to mask the broken copy. Generally speaking, the recessive allele contains a mutation where it does not make a functioning protein, so it makes a malfunctioning protein. So let's say this white allele has a nonsense mutation, which means that, the protein synthesis stops too early, right? The protein doesn't work. So the enzyme that it's supposed to make that makes purple pigment no longer functions anymore. Okay? If you're not making the purple pigment, your flower looks white. So as long as you have one of the dominant alleles, you're making the enzyme that makes the purple pigment, your flower ends up purple. I think that's an important thing to kind of think through. So, vocab. Genotype, first phenotype. This is the difference between how an organism looks and which alleles it has. So in our flower color, for example, phenotype was either purple or white. So it's a physical trait, description of an organism's trait. Yellow or green peas, wrinkled or round, all those traits are phenotypes. Genotype tells us which alleles it has. So does it have the dominant allele or the recessive allele? Two dominant alleles, two recessive alleles, one of each. So we have some of Mendel's results over here. Go ahead and take a couple seconds in your groups to discuss this F1 generation in terms of dominant and recessive traits, dominant and recessive phenotypes, and then phenotype versus phenotype. Take a couple seconds to talk about that. How do we do it?
Definitely have a dominant trait because the purple masks blue. And it's physical. Yeah. I haven't said that in so long. We better. Well, that's
Okay, so phenotype is a trait. So in this case, we mixed one parent that showed the purple phenotype and one parent that showed the white phenotype, and all the F1 offspring ended up having the purple phenotype. If we think about genotypes, we're dealing with a true breeding purple flower. What do you think its genotype probably is? Dominant. Yeah, both dominant copies of the. This plant is white, showing the white phenotype. What genotype do you think it probably has?
Recessive.
Both recessive copies. Why do you know this has to have both recessive copies? Because if it had a dominant allele, what color would it be? Be purple. Okay. This offspring here, we can't, well, we do know if we have these, this parent's going to pass on the dominant one in every gamete. This parent's going to pass on the recessive one. So this individual actually has one of each, right? It has the genotype of one of each. But the phenotype is still purple. So there's a difference between, important distinction between genotype and phenotype. Okay, another review of terms. Homozygous versus heterozygous. The prefix homo means same. So in that case, if they're homozygous, they have the same allele. Either homozygous dominant, both dominant, or homozygous recessive, both recessive. Two of the same. The prefix hetero means different. So heterozygous means a zygote that has different alleles. Capital P, lowercase p, one dominant, one recessive. So if we take a look at Mendel's pea plants again, how many potential genotypes do we have? Three, right? Three different genotypes. Homozygous dominant, homozygous recessive, or heterozygous. How many different phenotypes do we have? Two. Purple or white. Okay, dominant or recessive. So again, there's a distinction between what genotype you have and what phenotype you have. Okay. If we think back to proteins, as long as you have a dominant allele, you're going to make the purple pigment. If you don't have a working copy, you can't make the purple pigment. You end up being white. All right, there's some practice here on your handout. I know some of you might have worked ahead and done that. Just take a second. So these are all genotypes, right? They're showing which alleles you have. So homozygous or heterozygous. And compare with your classmates when you get that done. Again, as long as you have two of the same, it's going to be homozygous. So whether it's recessive or dominant, okay, we're going to be homozygous. If you have one of each, one dominant and one recessive, then it's going to be heterozygous. Okay, so same alleles, homozygous, different alleles, heterozygous. For each of the genotypes, what would the phenotypes be?
That was a trick question.
Share those with your group members like that.
What a tool is this trick?
This hopefully becomes something as like automatic as your times tables, right? So we're getting into more advanced genetics here. Like this is something that just needs to be like boom, boom, boom. As long as you have one dominant allele, you're going to show the dominant phenotype. So these two are purple. If you're homozygous recessive, that's the only time you show the recessive trait, right? So, you know, brown eyes, blue eyes, round seeds, regular tails, heterozygous and homozygous dominant individuals have the same phenotype. You have to be homozygous recessive to show the recessive phenotype. You have to have two broken copies to not show the trait. So are y'all feeling pretty comfortable with this so far in terms of everything? Okay, I just have a couple more slides. So when we do, and we kind of already talked about this, when we do make crosses, we can use alleles as letters. So again, dominant, purple, recessive. Actually, technically the correct way to assign these letters is using the recessive trait. So this is actually not the correct way to assign. Generally, we would use W's in this case. Usually, whatever the dominant trait is, that's what your letter should come from. But in this case, we're using P. So capital means dominant, and lowercase means recessive, right? So homozygous dominant. If we're a true breeding purple flower, we're going to have both dominant copies. If we're a true breeding white flower, we're going to have both recessive copies. Right? So we already talked about this. Anytime you cross a true breeding dominant with a true breeding recessive, all the F1 are going to be heterozygous, right? And that happened in every single trait that Mendel did. Anytime you cross a pure dominant parent with a pure recessive parent, you end up getting a heterozygous individual. And what trait will a heterozygous individual always show? The dominant one. Okay? And that's why the F1 generation was always showing the dominant trait, because they always got the dominant version from the dominant homozygous parent. So if we go back to this diagram and we fill in the actually you fill in the genotypes. So take a couple seconds, write the genotypes next to each one of these flowers. What are the genotypes going to be?
Oh, no, this is awesome.
Purple parent should be homozygous, dominant. Pure breeding recessive parent should be homozygous, recessive. All the F1 are going to be heterozygous. Okay. They got the dominant copy from one parent, they got the recessive from the other. This white flower offspring has to be homozygous recessive. What about these offspring? Can we know for sure? No. Okay. They could either be homozygous dominant or heterozygous. Okay. The ratio should be, 1 to 2. If you think about the Punnett square, we haven't gotten to that, reviewed that yet, but we don't know just by looking at them whether they're homozygous dominant or heterozygous. Yeah, they're really easy. Okay, so this is actually on the back side of your worksheet. So talk through these questions with your group really quickly.
What training key points are you studying the cross above the height? Where are the two alleles of this trait? Dominant and recessive trait. Which is dominant? Tall height. No, not short. Which is recessive short. And what alleles do? They have the what's a capital T and lowercase t, okay?
So we got enough. You need a little more time. Ready? Okay. So the trait we're dealing with here is height. The two different versions are tall and short or tall and short. Okay. And so those are also then going to be the alleles, right? So we have these letters. As long, the most important thing is you specify which letter is which. So technically, you would use the first letter, the recessive trait. Okay, so if you use T's, it's fine as long as you specify. I'm going to say tall, which is the dominant allele, and dwarf, which is the recessive. I know tall is dominant because, yeah, the F1 generation is all tall, right? So the dwarf or the short version is the one that got covered. The short's recessive, the tall is dominant. The F1 generation are going to have what alleles? Yeah, they're going to be heterozygous, right? They're going to get the tall allele from this parent, and they're going to get the dwarf or short allele from this parent, okay? So homozygous dominant, homozygous recessive, these individuals are all going to be heterozygous, okay? This genotype is going to be homozygous recessive. These can either be homozygous dominant or heterozygous, right? So we don't know for sure. So sometimes what we do is this. We draw the dominant allele and then we draw a dash to represent that could be either a dominant or a recessive. I didn't get to this in the other class. We have time. So this is the first quick check. One. This is real simple.
Oh my God, goodness gracious.
We're gonna have a lot of paper quick checks. It's hard to replicate online.
Writing out the.
Yeah, I got it square up.
What letters do
Huh?
what letters do
I mean, like I said, the general rule is to use the first letter of the recessive tree. But as long as you specify, then as long as your dominant allele is capital and your lowercase is zero, recessive still keys. So just follow through the questions at the bottom there. Don't start filling in lines until you know what you're supposed to put there.